All right, guys, welcome to 7.3. In this video, we're going to look at dihybrid crosses. Let's jump into it. Okay, so a dihybrid cross is when you have a cross that involves the alleles of two different genes. Okay, so now we're interested in knowing, what kind of offspring you're going to get if you have an individual with this trait and that trait, right? So, for example, if we look at Mendel's P's from our first example in 7.1, we can perform a dihybrid dihybrid cross to predict the outcomes of both a trait like, for example, pod color and pod shape. So what are the chances of, green pods and inflated or full pod shapes, right? So let's say, for example, right, capital G is green pod, lowercase g is yellow pods, right? So green pods, yellow pods over here. And then you have a second trait that we're investigating, which is pod shape, that it could be capital F for inflated or full pod shape, and then lowercase f for a constricted or wrinkled or flat pod shape, right? So now we're interested in both of these traits being mixed up together, right? This also means that you're now going to have 4 gamete combinations and not 2 for this particular individual. Why? Because you got 2 traits and then there's two alleles for each trait, and therefore you have 4 allele combinations, right? So for example, right, let's take one particular plant, and the plant has this particular genotype, G, capital lowercase g, capital F, lowercase f, right? What are the phenotypes? Well, if we are assuming a complete dominance, which we are, it's going to have green pods because of this green pod allele, and it's going to have inflated or full pods as well. So if that particular plant was going to develop pollen or ovum, you're going to get 4 different possible combinations. So you're going to get this capital G together with this capital F, which is this one here, this capital G with this lowercase f, which is this combination here, this lowercase g with a capital F over here, and then a lowercase g with a lowercase f over here, right? So you're mixing the G's and the F's together until you have every possible combination. So now you have up to 4 different combinations for this particular individual. Now, let's say we have another individual, this one here, lowercase g, capital F, lowercase f. So this plant here, they're going to have yellow pods that are inflated, right? So capital F there for inflated. So yellow pods that are still inflated, and so their genotypes, what are the possible genotypes? Well, if you mix all the G's and F's together, you have lowercase g, capital F, which is this one here, lowercase g, lowercase f, which is this one here, lowercase g, capital F again, which is over here, and then lowercase g, lowercase f. So now you only have 2 possibilities, right? Which is only 2 gametes on this particular section, right? So that's kind of how you differentiate. So every time you see a dihybrid cross with this kind of two different notations here, you have to remember that you have to form 4 different possible gametes. It also means that your dihybrid Punnett squares can be quite big. It can be up to a 4x4, right? So to accommodate for all your possible combinations. It could be less, like, so for example, in that previous example, was because there was only 2 for the second one, it would have been only 8 squares long. But in this case here, it's going to be 16 squares, right? So let's go with an example. Let's go with Mendel's P's, right? So we're going to go with the same examples of green pods and flat or wrinkled pods, right? So let's say you had this individual, plant 1 with capital G, lowercase g, capital F, lowercase f, right? Which will have green pods and inflated pods, right? Mixed together with plant 2, which is the same, right? Capital G, lowercase g, capital F, lowercase f, right? So you're going to determine all of the allele combinations for plant 1 and for plant 2. So you're going to have, this is all the possible combinations, G, F capital, G, F lowercase, lowercase g, capital F, lowercase g, lowercase f, and then you're going to see the same here. So if you map them out, that's what you're going to get. So here are all the possible combinations along the top and along the side for these 2 plants. And then from there, you just, once again, like a Punnett square, you just fill all the combinations together. And, this will have 2 capital G's, 2 capital F's. And if you just fill the whole thing out, you're going to get this as all your possible outcomes, right? So all of these individuals, then you have to kind of read the genotype and make sure you get the correct phenotype. So, these individuals, they're all going to have green pods because of the capital G. here, 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 all along here, and all along here as well. And then if you have a capital F, they are going to have full as well. So all these individuals here, 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 and then this individual here and here, because of the capital F, are going to have full pods. If they have lowercase g, they're going to have yellow pods, and if they have capital F, they're going to be yellow and full. But then if they have yellow and lowercase f, they're going to be yellow and wrinkled, right? So now you got a whole host of different possible results that you could possibly get. Now, if you were to analyze the cross, your genotypic ratios, there would be way too many. You have like 16 different combinations in this particular case. Actually, probably a little bit less. I think it's like 13 different combinations because some of them repeat themselves, like these two guys here repeat themselves here, and then same with like maybe these two guys here. So, you know, you got a lot of different ratios, right? However, your phenotypic ratios can be plotted a bit easier, right? So, you know, how many green inflated do we have? How many green constricted do we have? How many yellow inflated do we have? And how many yellow constricted do we have? They have to match 16. So eventually you should get a ratio of a 9, 3, 3, 1, right? 9 individuals are, you know, green and inflated along here, and these two guys here. 3 of them are green but constricted here, here, and here. And then 3 of them are yellow but inflated. And then 3 of them are yellow and constricted. You can see this is the double recessive down the bottom here, right? So that's a dihybrid cross for you know, for punnets. You know, you can do with any other examples that you would like. I'll leave it to you guys to actually work that out yourselves for other traits. Now, one of the key things you need to distinguish in a dihybrid cross is whether or not the genes are linked or unlinked, right? Unlinked genes means that both the genes are found on different chromosomes, right? Because they are on different chromosomes, they're going to be sorted independently of one another during meiosis, which means that they're going to appear in pretty much equal ratios, right? However, if they are linked genes, the two genes are on the same chromosomes, which means most likely they're going to appear together with a very small chance of being separated due to crossover and recombination and the shuffling of genes during meiosis, right? So linked genes affect the chances that the two alleles are going to be passed on either together or separately, right? So with unlinked genes, you denote them with a semicolon here, right? And then with linked genes, you denote them with a slash, right? And so if you see this little indication, most likely they were referring to unlinked genes that are going to be sorted independently, and you don't have to worry about the ratios and the numbers of offspring. However, if you see a slash between the two, it's most likely going to be linked, and you will need to pay attention to the number of offspring and the ratios there, okay? Now, let me go with an example, right? So let's go with linked genes for rhesus blood type and elliptical blood cells, right? So rhesus blood is determined by the RHD gene, and if you have a capital D, you are rhesus positive. If you have a lowercase d, you are most likely going to be rhesus negative, right? Likewise, that's a complete dominance trait, by the way. And then likewise, if you have a capital E, you are likely to have elliptical blood cells. And then if you have a lowercase e, you will have normal blood cells. Now, both RHD and EPB41 genes are located on chromosome 1, and therefore they are linked genes. This means that during meiosis, you get a small chance of the recombination occurring and separating the alleles, right? And so this is just a diagram to show you, this is what rhesus positive looks like, and rhesus negative has nothing. And then elliptical will kind of have these kind of this is uniform and this is symmetrical, whereas elliptical will have a kind of a bend in it. Okay. Now, how do we understand that? Well, if we looked at linked genes for rhesus blood and elliptical blood, let's say you have two individuals. Let's say Sarah is heterozygous for both traits, right? She will have capital D, lowercase d, capital E, lowercase e for those traits. You will need to denote them with a slash in between now that we know that it is a linked genes. And then Dave will have, he's going to be homozygous recessive for both traits, right? Lowercase d, lowercase d, lowercase e, lowercase e. And it's also denoted with a slash as well. Now, the possible allele combinations for Sarah, if you mix them up, you will get DE capital, DE lowercase d capital E, and then 2 lowercase e. David, on the other hand, that's meant to be a lowercase d, sorry. He's only got one possible combination, lowercase d, lowercase e, right? So once you have that, you can map out the Punnett square that's going to show. Here's a diagram to show you what they will look like. So Sarah, this is what she's got, right? One of her chromosomes have this, one of them has this one, and then, they're both found on the same chromosome, right? Dave, on the other hand, he's got exactly the same, and they're both found on the same chromosome as well. And if you mix them together, you don't get a 16 square Punnett square, right? You only get a 4x1, right? Why? Because Dave has the same genotype. So he's only got one possible combination, right? She, on the other hand, she's got 1, 2, 3, 4 different combinations, right? So once again, all the different allele combinations are there. However, if we look at the percentage ratios, they're all going to be different. Why? Because they're on the same chromosome, most of the time they're going to be sorted together. So you're more likely to get two capital D, capital E, or lowercase d, lowercase e, up along here and along here. In fact, 0.49 out of 1, so 49% of the time, you're going to get red, capital D, capital E, or green, capital D, lowercase e. And on a very small chance, 1% of the time, you're going to get the other two combinations. So if they were to have kids, most likely their kids will have rhesus positive and elliptical blood, or rhesus negative and normal, but the in-betweens are very unlikely. Okay? So that tells you that these genes are linked because their offspring ratios are different. If it was independent, it should be 25, 25, 25, 25, right? But it's not. It's 49, 49 to 1 to 1, right? So that's why it tells you that it is linked genes, okay? Now, when we are referring to the eggs in this particular case, these are your parental eggs or non-crossover eggs, right? So these are the non-crossover ones, and therefore they're the most likely to occur. And then the percentage ratio is written here as 0.49, but you can just assume it's 49%. And then these ones here, they're the recombinant eggs. Why? Because in these examples here, the eggs have recombined in their gene segments and given a segment to each other. And therefore, the smaller percentage ratio are always going to be the recombinant ones. Okay, so that's a linked one. If it was unlinked, you would treat them as per normal and everything will be 25%. Now, a dihybrid test cross is designed to, slightly differently from a monohybrid test cross. In the monohybrid test cross, we are trying to figure out whether an individual is unknown or not. However, in a dihybrid test cross, we're actually trying to figure out whether or not the genes are linked or not, right? So a cross between a double homozygous recessive for both traits and a double heterozygous genotype for both traits is a standard dihybrid test cross, right? So the idea behind it is you already know the genotypes of the parents, but you're trying to figure out whether that particular trait combination is going to be linked or unlinked. And so let's say the first parent can only produce lowercase b, lowercase f gametes, whereas the second parent are going to produce, all four of the possible combinations because they are a double heterozygous one, right? So in this case, we're going to use hamsters, guinea pig fur, right? So I've got pictures of guinea pigs here. I don't know why I said hamsters. The capital B is going to be black fur versus lowercase b for white fur, and then capital F is going to be shaggy fur, which is this is what shaggy is meant to look like, versus short, right, which is lowercase f, right? So if we do the dihybrid test cross and the offspring ratio is 1 to 1 to 1 to 1 for all the traits, then therefore the genes are unlinked. However, if the ratio isn't, we can assume that the genes are linked. Okay, so here's an example, right? So let's say you've done your test cross, and there is your double homozygous one with your double heterozygous with your different gamete combinations, and it comes out like this, right? Long black, long white, and short white for the combinations. Okay. If it was unlinked, you should be expecting this in equal proportions, right? That tells you that they were on different chromosomes and it was just sorted independently of one another, and you didn't have to think about it like just a normal shuffle. Oops, and oops. There we go. So the dihybrid test cross can also be used to figure out how closely linked they are as well. Okay, so that gives you an indication of whether or not the genes are, you know, really close together or actually a little bit further apart from one another in terms of their distance, right? Pretty remarkable if you think about it, right? So how do we figure it out? Well, it's actually quite simple. Distance between the genes is actually calculated by going 100 times the number of recombinant offspring divided by the total number of offspring. Basically, what's the percentage ratio of the recombinant offspring, and that's how many map units apart they are from one another, right? So for example, let's go with an example here. So you got Shaggy and Honeypots, right? Let's say Shaggy is our individual along the top here, and then Honeypots is the individual with the double homozygous, right? And then the combinations appear like this. 7 of their offspring are going to be long black, 2 are short black, 2 are long white, and then 7 are short white. Now, because of that imbalance of possible ratios, that tells you that most likely these are linked genes, right? They're linked because of the difference. Now, your parental offspring, so your parental eggs are this one here with the BF capital and then BF lowercase, right? Why? Because they're the majority, right? And then the recombinant are these ones here, the 2 and the 2 here. So I can take that percentage and I can actually calculate out of how many that is. So there is a total of 7, 9, 11, 18 offspring, and 4 out of the 18 offspring are recombinant. So based on that, I can calculate that. So I can use my calculator and I go, well, what's 4 divided by 18? That's 22 times 100. So yeah, 22 map units apart. So the distance between the loci of the genes is about 22 map units. Why? Because there is a 4 out of 18% chance of the recombinant offspring occurring, right? So here's another example. So for example, let's say you've got parent 1 with this particular test cross here and parent 2 with this one here. And let's say it's a 44%, 44%, 6 and 6%. So the total ratio of recombinant is 12. Therefore, the number of map units apart from one another is going to be 12 units, right? So the smaller that percentage is, the closer the genes are linked and the less likely they are to be recombinant. The further apart they are, the more likely they are to be recombinant, okay? All right, so that's it for dihybrid test cross and for chapter 7. So I'll see you guys in the next chapter.